yes. Hi, everybody. It's a real pleasure to be here. And so today I'm going to tell you a little bit about what's happening in the world of harnessing human immunity to treat emerging viral infections, with a special focus on hantaviruses, which I know are an important public health problem here in Chile. And as the host mentioned, I'm a professor of microbiology at Albert Einstein College of Medicine in New York City. And my group of 15 people really work on emerging viruses and how they cause disease and what we can do to deal with this huge emerging problem. So this actually, this little map was drawn by hand by Dr. Anthony Fauci at the NIH, just to list all the different emerging viruses that were sort of popping up. And not just viruses, but also bacteria and other kinds of microorganisms that are jumping into people from animals. This is a very incomplete list. It stops at 2017. And of course, we know that something really big happened, very soon after that with the COVID-19 pandemic, which really showed the whole world how important it is for us to really think about these viruses and other microorganisms, bacteria, that normally just go about their business infecting animals, but every once in a while they jump into people in mysterious ways that we don't really understand, and we have to deal with them and the consequences of them jumping into human populations. So what you're seeing here is just a list of some of the viruses that have jumped into people or have sort of jumped again after being gone for a while, back into humans. And you can sort of get a sense that the trend is accelerating. This is starting to happen maybe a little bit more quickly. And that's interesting, and we don't really completely understand why that is, but we can take some guesses, and I'll sort of talk to you a little bit about that. So certainly, we're starting to understand a little bit better how viruses emerge and cause disease in humans, but it's a really complicated problem. There are a lot of different factors. Chance plays a really big role. So we know that viruses or bacteria can infect animals, and then every once in a while they can jump into people. But then something mysterious happens, which is in the middle here, the orange circle, where somehow the virus has to mutate and evolve, and then find a way to jump from person to person to person and start causing disease, just like the SARS-CoV-2 did with the COVID-19. And then, of course, it can spread globally, causing a big outbreak or even a global pandemic, as we are still in the middle of. So one thing that we're starting to understand is that certain kinds of animals, certain kinds of mammals, are really important hosts, what we call reservoir hosts. So these are where a lot of viruses live. They just are normally doing their infections, and they're there. And the animals are generally okay with these infections. That's just part of their life. But every once in a while, these viruses can jump either directly into human beings, or they can jump into some other kind of animal, maybe like a camel or a pig or a cow, and then from there it can jump into people. And so this is just a slide showing you what this looks like for many different coronaviruses, like SARS-CoV-2. But this is generally true for emerging viruses, that can jump into people. And what you'll also notice is that rodents, like mice and rats, and bats are really, really important hosts for many viruses that come into people. And this is just partly because there are lots of different kinds of rodents and bats compared to lots of other kinds of mammals. So these are just very common mammals, and we often come into contact with them, and that increases the chance that these viruses can jump into us. And of course, another really big factor in causing viruses to jump into people and then to spread globally is how human populations have changed and how human connectivity has changed just in the last few decades. So on the left, you're seeing the human connectivity through airplane flights in 1933 on the top. And on the bottom, you can see what that looks like in 2020. So this giant bowl of spaghetti is humans traveling every day from one corner of the globe to another, just as many of us came to this conference to be able to speak and attend. And of course, as we know from COVID, this is a great way to spread viruses very quickly. And then on the right, what you're seeing is climate change. So global temperatures in 2000, and then what are being projected as global temperatures in 2100 and sort of beyond. And so as the climate changes, animals are moving, plants are changing, and this is changing how we interact with our own environment and how we come into contact with different kinds of animals in the world, which could potentially cause us to, become exposed to viruses that they normally carry. Okay, so this is sort of a general thing, but today I really want to talk to you a little bit more specifically about hantaviruses. So I think for a lot of people in Chile, hantaviruses are well known, but they are actually an emerging global problem. So this is what, on the left, the outside of a hantavirus particle, or like what we call a virus particle, looks like. And this is like a suitcase that's carrying the genetic information of the virus, which is shown here on the right. And this genetic information needs to be taken into the cells of your body. It reprograms the cells and turns them into virus factories. And that's how the disease starts. And so the job of this outer shell or suitcase of the virus, which is just as beautiful, for those of us who study it, we find it to be really elegant how it's engineered. It's a structure that really is designed to keep the genetic information of the virus safe, but then also to bring it into the cell of the host at the right time in the right place to then take the cells over and then eventually take the host over. So if we're going to develop vaccines and treatments, a lot of times we're focusing on this outer machinery of the virus, the suitcase of the virus, to find a way to keep it from opening, to keep the genetic information inside the virus, and to prevent the virus from taking the cells over. So that's what hantaviruses look like. And these are the kinds of animals that they can infect. You can find hantaviruses on every continent except Australia and Antarctica. And they infect a really wide range of animals. Everything from rats to moles to shrews, and even some really strange-looking fish over here on the side. This is called the mini pizza batfish, and because it looks like a pizza, and it has a hantavirus. But there's lots of hantaviruses, but there are actually only a few that cause disease. And the virus that causes disease here in Chile and then in Argentina is called Andes virus. And this is actually a newspaper clipping from a case in 1997, which was this big outbreak of hantavirus in the southern part of Chile. And so, in fact, hantaviruses cause two different diseases. In Europe and Asia, it causes a disease called HFRS, which is primarily a disease of the kidney. And in North and South America, it causes a disease called HCPS, which is primarily a disease of the lung and the heart. And so what we see with people who get infected with Andes virus and get really sick is that their lungs fill up with fluid. So this is a sort of a chest X-ray, and the left lung, which is on the right here, looks fine. But if you look at the right lung, you can see the bottom part, you can't even really see it, and that's because it's full of fluid. Because the lungs are filling up with fluid, and so people can't breathe anymore. And this is why they really get sick, and many of them die. And the reason this happens is because the virus has evolved to infect the cells that are lining the blood vessels. And so what you can see here on the left is a relatively normal-looking section of somebody's lung. And what you're seeing on the right is a lung from somebody with hantavirus infection. And you can see that here there's a lot of white, and that's air, because that's what lungs are supposed to do. They're supposed to take in air and exchange carbon dioxide for oxygen. But in the right, you can see that the lungs are full of liquid. And that's because the cells are infected, and they're releasing all this liquid. And so the person really kind of drowns in their own fluid that they're producing in their lungs, which is what makes this infection so dangerous. And the way you get hantavirus infection, it doesn't matter if you're in Sweden or in China or in New Mexico in the United States or in Chile, you really get it by coming in close contact with a mouse or a rodent that carries it. And you breathe in some material from the mouse, and then it goes into your lungs, and then you can get infected. And so, in fact, a lot of work has gone into understanding exactly how this happens in Chile and Argentina for Andes virus. And the scientists who've worked on this have identified this little rat as the carrier of Andes virus. And every once in a while, in every couple of years or so, there's a blooming of this Chilean bamboo called quilihue. I hope I'm saying that right. And when this bamboo flowers and produces seeds, the rats eat it. And then there are many, many, many more rats because there's lots of food. And then they come into contact with more people, and so more people get hantavirus infections. And this is sort of an example of the ecological dynamics that we see with these kinds of rodent-borne infections that humans can get. And by understanding these things, which requires people to go in the field and collect these rodents and sample them and really sort of try and predict where the outbreaks are happening, we can really prevent these outbreaks from happening, although it is quite challenging to do that. And then, of course, public education is a very important aspect of this. So these posters that are actually real posters from different places in Chile are really kind of trying to make the public aware of the dangers of hantaviruses and how to protect themselves when they're out in open spaces or camping or cleaning out their sheds or whatever they're doing that could bring them in contact with rodents. Okay, so we can do a lot to sort of educate the public, to prepare doctors, and then to also do prevention. So what can we do, ways to reduce the risk of infection. But ultimately, no matter what we do, people are going to get infected. And when they do, what can we do to help them? So what can we do, what can we give to people who are already infected and are at severe risk of disease? And so what we and many others have been working on for hantaviruses and for other emerging viruses are these beautifully engineered machines that our own immune systems make. Our own immune systems are among the greatest drug designers on this planet. And you have an ability in your own body, when you're exposed to a virus or any kind of bacterium or any kind of microorganism, to make special molecules called antibodies, which are these Y-shaped molecules here. And they can very specifically recognize the structures on the surface of the virus, on the suitcase, and block it. And so, that's really what we're after. We don't want to make new drugs. We want to find the drugs that people who've been exposed to hantavirus infection already make. And so we already know that if we take blood from somebody who has had hantavirus infection and survived, that we can give that blood, which is full of antibodies, to somebody else, and it helps them. And so this is actually work done by one of my collaborators, Dr. Pablo Vial at Universidad del Desarrollo here in Santiago. And they're able to show that this actually works. But it's not that easy to do. It takes a lot of there are not that many samples of blood available, serum available from people, or plasma. So what we really wanted to do was to take this huge collection of antibodies that's in the blood of a person that's been exposed to hantavirus and find that one special antibody that does what we want. We don't care about all the others. We just want that one. And this is a little bit like going into an enormous library and looking for a single book, right? But amazingly, the technology to do this exists now. And we've actually worked with a huge group of scientists from all over the world, from Sweden, from the United States, from here in Chile, to kind of get blood from people who have had hantavirus infection and find these special magic, we like to call them rainbow unicorn antibodies that we can give to people. And we call this consortium Prometheus. And some of the critical people here are here in Chile, Dr. Cecilia and Pablo Vial, as well as in Sweden and New Mexico and the United States. And they are the ones that are the doctors and the clinicians who are getting us the samples from people that we can use, the libraries, if you want. They're giving us the libraries from which we can find the books from the single antibodies against hantavirus. And so we actually found one such antibody. And so what you're seeing here on the left is this one antibody binding to the surface of the hantavirus. And that's just looking from the side, and this is looking from the top. So this antibody has been made by somebody's immune system to bind very beautifully and perfectly in just the right place. And when it does that, so normally what would happen is that the inflammation of the virus would come out, and the virus would infect the cell. But now these antibodies can bind, and they can prevent this from happening. And so now the virus is stuck. The virus cannot unpack, and so it can't really go on vacation because it can't unpack. And that's kind of how we think about this. This is the virus with its weapons, and here's our perfect countermeasure that's evolved just right by your own immune system to kill the virus. And so we just show very quickly here that in an animal model, which is a Syrian hamster, if we normally infect with the virus, the animals all die within a week. But if we give them the antibody, within 2 weeks, if we give them an antibody at 6 or 7 days after they've been exposed to the virus, most of them survive. So here you're just looking at survival. The higher the line means the more of the animals survive. So the antibodies work. They can protect against animals. But can they protect against people? And what would it actually do to get these antibodies into people? And why is it so hard to make such antiviral treatments available to people when and where they're needed, right? And so this is what we sort of refer to in the field as the valley of death. Here we are on the basic science side, and we need to get over to this side where we actually need to produce a lot of antibody that's good enough to go into people and safe enough to go into people. This takes studies in animals, which is expensive, but can be paid for by the government or by nonprofits. It takes, really importantly, clinical-grade manufacturing, being able to produce large amounts of this antibody that's clean enough to go into people, which is incredibly expensive. Usually companies do this, but for something like hantaviruses, it's very hard to convince companies to do it because they're not going to make any money. And then, of course, you have to be tested for human safety, which is expensive. And then there are all kinds of regulatory hurdles that have to be overcome, which also costs money and takes lawyers. And of course, importantly, there's intellectual property issues. So we need new models to handle these emerging infections. And I'm just going to finish by telling you quickly about one model that our consortium has, which is the company that's developing this treatment is giving the antibody for free without any intellectual property strings to the 3 clinicians in Chile, in Sweden, and in the US, so that they can run clinical trials to test these antibodies in people. But of course, that's not enough. We need to have these antibody treatments ready ahead of time, and we need to move them through human safety trials without needing the full we don't need these to be approved drugs, we just need them to be shown to be safe in people. And we need to, most importantly, decentralize manufacturing. And this is also something where Chile has taken important steps. Dr. Alexis Keller, who's here in Santiago, and many of his colleagues have worked together to build a manufacturing facility for vaccines and antibodies, and we're going to work with him to try to get our hantavirus antibody made here in Chile for local use, which is what I think we really need to do in the long run. And then we need to sort of work on the regulatory issues, and we need to figure out how to deal with intellectual property to make these things available during an outbreak or before an outbreak where these drugs are needed. And so we need to find new ways to pay for all of this. It can't just be companies. Governments have to step in. Nonprofits have to step in. And philanthropy has to step in. So global problems require global solutions, but they also require implementation of these global solutions at the local level. And that's the only way we're really going to deal with these emerging infections that, I can't tell you when the next one's going to happen. I can't tell you which virus it's going to be, but I can tell you there is going to be one, and probably more than one. So, the only way we're going to get ready for this is by doing it together, by implementing global solutions and finding a way to decentralize a lot of the aspects of this so different regions can work on their own to deal with their own local problem as it occurs and when it occurs. So with that, I'll stop, and thank you very much for your time. Thank you.
Thank you.